All right, everyone. Well, hello. We're now on chapter 10. Chapter 10 is kind of a bunch of different things. And I'm going to use this opportunity to cover energy systems. Many of you remember this fondly from your biology classes. And we're just going to cover a bunch of things that really will help us understand different systems later on. So we're going to talk about energy systems, hopefully make this as painless as possible. I literally have been teaching this in my other bio classes, so this is like the 50,000th time, if that's a word, that I've been teaching this. All right, so anyways, we're going to start with energy systems. Let's go. So there is a question of how do cells make energy? So now would be the time where I start talking about plant cells, but I'm not, because this is not a class where you really care about plants. So anyways, we are what is called a heterotroph, meaning that we must consume organic molecules to make energy. Now this is different from plants. Plants are what we call autotrophs. So basically what plants do, all right, so I told you I wouldn't talk about plants, but here I am talking about plants. So anyways, plants are what you call autotrophs. They can make organic molecules from sunlight, water, and carbon dioxide. Now, many people think that, well, if we could do that, that would be super cool, right? No, it would mean that if you go out suntanning, you possibly could become obese. You could gain weight just by sitting outside and getting a tan. So, anyways, okay, so sorry for the pause there. So anyways, we need organic molecules. And with us, we have different options. We'll talk about the options later. But we really prefer a sugar molecule called glucose, which is actually the simplest sugar that our body uses, our cells use in order to make energy. Now, basically, what's going to happen is that glucose is going to be put through a series of steps that we call cellular respiration. And more or less what we're doing here is we are taking the energy, the potential energy stored in glucose, we're converting it to an energy molecule called ATP. Or if you want the more formal name, adenosine triphosphate. Okay, so it's this molecule here that we are focusing on right now. And what exactly is ATP? Well, I guess we can get really involved in it, but let's just try and keep it simple. Just make sure I have the outline. Is this molecule looks like this. So here we have adenosine triphosphate. And adenosine triphosphate is basically, it is the high energy form of the molecule. And it looks like this. It is in adenosine side group combined with 3 phosphates. Now, what is this phosphate business? Well, a phosphate is a molecule that looks like this, in case you're curious. It's a phosphorus with 4 oxygens attached. Now, each of these oxygens has a plus, or sorry, not plus, has a minus charge to it. Now we know that, or we don't know, maybe you will know now, is that molecules or atoms or elements, whatever, that have negative charges, they repel each other. They don't like each other. So they're trying to get away from each other. And these phosphate groups with their negative charge, they're bound together, but they don't want to be. So with a very little motivation, what can happen is that through an enzymatic process called hydrolysis, is this third phosphate can be cleaved off, and it's going to burst away. It's going to fly away, and it's going to release energy. Because this third phosphate is really trying to get away, and if you just give it a little bit of motivation, think of holding a big dog on a leash, and the dog's trying to attack a squirrel or something, and you're holding onto the leash, and the dog's tugging against it and tugging against it. Okay, let's just roll with it. And the dog is tugging so hard that it eventually breaks the leash and goes flying off. When the leash breaks, it's going to release energy. All the energy stored in the leash is going to be released when the leash is broken. That energy generated is similar to what happens with ATP. When that third phosphate is, when that bond is broken, it's going to explode off, so to speak, and it's going to release energy. And this is a form of energy called kinetic energy. Now, what is kinetic energy? Well, it is energy in motion, meaning that it is energy transformed into something that creates movement. For example, if gasoline is going to be potential energy, combusting the gasoline to turn the engine is going to be kinetic energy. ATP allows us to do kinetic energy. We'll talk about more of that later. Now, the molecule has a low energy version, and this is called adenosine diphosphate, also known as ADP. And what this is it is an adenosine side group with two phosphates. And with this one here, the two phosphates, they're trying to get away from each other, but not that much. There's not much energy stored in this bond here. So breaking off the second phosphate is not going to give you too much of a payoff. You have to keep in mind that it takes a little bit of energy to even break this bond. So it would not be worth it to use that bit of energy to break this bond. It would actually just not result in a suitable payoff for the body. So what happens here is that our body is constantly taking, or our cells, are constantly taking ADP, and they're going through an enzymatic process. So it's going through a process called cellular respiration, which is actually a very long process, as we'll find out. And we're going to make it adenosine triphosphate, or ATP. So it's constantly being cycled between the high and low energy versions. Okay, so ADP results from when ATP is used for energy. So once again, maybe I wasn't clear on this, is glucose is our body's form of potential energy. Potential energy means that it has the potential to be converted to kinetic energy. So this is going to be potential energy. And kinetic energy is ATP. So basically, we're transferring the energy in glucose toward the kinetic energy in the form of ATP. Okay, so I'm going to draw a cell here. And we'll just say that this is a muscle cell. So what's going to happen here is that cells are highly obedient. And cells have on them something we haven't really learned yet, but they have something called a receptor. And this receptor here is a special kind of receptor called an insulin receptor. And when the insulin receptor binds with insulin, it's going to receive the signal to take in a molecule called glucose. So body cells will not take in glucose without insulin. Brain cells, however, or brain cells, neurons, do not need to be told to take in glucose. They do so when they want to. But body cells, they need the okay from insulin in order to take sugar from the bloodstream. So in people that are diabetics, is they're not making enough insulin, not enough glucose is getting into their cells, and they have to make energy through a different way. Okay, so the very first step of making energy, and keep in mind this is the steps of cellular respiration, and cellular respiration is the process of taking glucose and converting it into ATP, or energy. Happens in varying stages. So the very first stage that happens as soon as glucose gets into the cell is a process called glycolysis, or glycolysis. Glycolysis. Actually, I wrote this too big. Go ahead and make this a little smaller. So the process is called glycolysis. Glyco for sugar. All right, sorry for the pause. I got a phone call. My mom wanted to say goodnight. All right, so yes, so we are at glycolysis, and so this is the very first stage of cellular respiration. And what happens here is that you're going to have a molecule of glucose, and it's going to be split into two molecules of what we call pyruvate. But there are a couple of steps that we need to talk about. So glucose is going to be converted to a form which actually costs the cell energy a bit, but we're not going to talk much about that, called glucose 6-phosphate. I know that looks like a G. It is indeed a 6. Glucose 6-phosphate. And this is important because it actually makes the glucose water insoluble, which is important for the next steps. And now glucose 6-phosphate is going to be converted into two molecules of what we call pyruvate. Pyruvate. Now, pyruvate is an interesting molecule. And it's interesting because, let me see how much yeah, I got to talk about the coenzymes. And pyruvate is interesting because its fate depends on whether or not there's oxygen. But anyways, so let me write this down for you. Chemical formula for glucose is C6H12O6. The chemical formula for pyruvate is C3H4O3. But recall there's two of them. So if you add them up, we have C6H8O6. Now, where did those other 4 hydrogens go? And I'll tell you. During the conversion from glucose 6-phosphate to pyruvate, we actually lost some hydrogens and some electrons, which is actually by design. And that's because, let me draw this here, the organelle, the mitochondria, is really where the majority of the ATP is made. And to do so, it really needs hydrogens and electrons. And as we're going to see as we go through this process, is more or less these chemical reactions are going to result in a loss of hydrogens and electrons. We just don't want these two things just floating around in the cell. So we have a coenzyme that I compare with to an Uber called an NAD+. And I compare it to an Uber because what it does, its job, is it carries hydrogens and electrons. Anytime hydrogens or electrons are released, it is going to be picked up by a coenzyme, like usually NAD+. And whenever it carries hydrogens and electrons, it's going to be converted to a new form called NADH. So NADH is what we call the reduced form of NAD+. You don't have to worry about what that means. Reduced just means accepts electrons. Just know that NAD+, when it carries hydrogens and electrons, converts to NADH. Then NADH is going to go to a very special part of the mitochondria called the inner membrane space. Anyways, we'll talk about that later. All right, so anyways, moving on to pyruvate. So pyruvate has two possible fates. Now, if oxygen is not present, if there's no O2, if there's no 2, I know, funny. If there's no O2, it's going to go through a process called anaerobic respiration. Anaerobic means without air, without oxygen. Anaerobic respiration. And the type that happens in animals is called lactic acid fermentation. So what happens is that pyruvate is going to be converted into ATP and a waste product called lactic acid. Now, usually your cells are well perfused with oxygen. They have lots of oxygen. However, let's say you're exercising really intensely and you're working your muscles so hard and they're using so much oxygen to make energy that they run out of oxygen. And you can feel this if you do very intense exercise. Like let's say you're doing sprints. At a certain point, you'll feel like your legs are getting tired, they'll start to burn, and then you will not be able to sprint anymore. Or if you're doing bicep curls, you're curling away, having a good old time, all of a sudden you start to feel your muscles burn, the reps become harder, and then you reach muscle failure. Well, what's happening there is that as soon as you deplete your muscles of oxygen, your body's going to switch to lactic acid fermentation. Now, this can make a little bit of ATP, but the trade-off is it makes an acidic product called lactic acid. And lactic acid is actually damaging to the muscles, which is one reason why when you work out really hard and you aren't used to it, you feel sore, and that's because of the damage done partially by lactic acid. And it also takes your body time to clear out lactic acid from the muscles too. But anyways, the reason why you start to feel tired is because when your body makes energy through anaerobic respiration, it can't make nearly as much. It's more of a backup pathway. Like it'd be really bad if you were running from a grizzly bear, then your muscles ran out of oxygen and you just face planted. That wouldn't be cool. So the anaerobic pathway lets us make energy even without oxygen. So we can keep on pushing just a little bit more or keep on running just a little bit further. So it's a very important pathway. But if we have oxygen, pyruvate is actually going to be carried to the mitochondria. So keep in mind, glycolysis and lactic acid fermentation happens in the cytoplasm. Let me write that here, cytoplasm. But the rest of the pathways that make loss of oxygen, they're going to happen in the mitochondria. So pyruvate has O2. Pyruvate is going to be carried to the mitochondria, and it's going to be converted into another product called acetyl-CoA. And as it's doing so, you are going to lose another hydrogen and electron. It's going to be picked up by NAD+. It's going to be converted into NADH. Then that also is going to the intermembrane space. Once again, in the mitochondria, pyruvate is going to be converted into acetyl-CoA. And yeah. So now we're going to go and, when it's converted to acetyl-CoA, you're going to lose some hydrogens and electrons. It's going to be picked up by NAD+ and converted to NADH, where it goes to the intermembrane space. So now we're going to go into the mitochondria. So now I'm just going to draw a big mitochondria. Yes, it is a square because I don't draw shapes too well. And the mitochondria actually has a double phospholipid bilayer. I'm draw like this. Very interesting organelle. So when we left our hero, we were at acetyl-CoA. And we have a cycle called the citric acid cycle. And it's a cycle because actually what's going to turn out is the last product made is the first reactant, but we're not going to talk about that. I did a job interview a while back where I had to teach about the steps of the citric acid cycle. I've never been so miserable preparing for a job interview in my life. Like that was mind-numbingly painful and boring. Okay, so anyways, acetyl-CoA goes into what's called the citric acid cycle. I'll write that here. Citric acid cycle goes by a couple of names. Also goes by the Krebs cycle, which is probably what you were taught in high school. So these are interchangeable. So what's going to happen is that acetyl-CoA is going to go through a series of chemical reactions that's going to release energy that's going to convert some of the molecules into CO2. So basically what's happening is that as acetyl-CoA is converted from one form to the next, it's going to lose carbons and oxygens, which it doesn't want anyway, so it just leaves the cell. And it's going to release hydrogens and electrons. So, and I'll go through this really fast here. So basically, if you think about it, is you are going to have this molecule here, that's the starting formula for glucose. And our cells don't really want the carbons or the oxygens. So when the citric acid cycle is when they start to remove those carbons plus oxygens, so they're only left with the hydrogens and the accompanying electrons. So what's going to happen here is that these hydrogens and electrons, they're going to be picked up once again by NAD+, and its helper, FAD2+. They're both the same thing, really. But I'd be teaching you wrong if I told you that it was just NAD+. There's slight differences, but basically they're the same. So NAD plus and FAD plus are going to pick up the hydrogens and the electrons, and they're going to be converted into their reduced forms, NADH and FADH2. Just recall that when I say reduced, it just means that they've taken on electrons. That's all. So both NADH plus FADH, they are going to be the version that's carrying the hydrogens and the electrons. And they're all going to the intermembrane space. So the citric acid cycle happens inside the mitochondria, called the mitochondrial matrix. What happens is that the modified pyruvate called acetyl-CoA is more or less going to be disassembled. So you get most of the hydrogens, most of the electrons, and the carbons and the oxygens are lost, combined, and excreted as carbon dioxide, and the cycle repeats itself. So now we're going to focus on this space here, the intermembrane space. And this process here, so this is going to be the intermembrane space of the mitochondria. And this form here is going to be called oxidative phosphorylation. Has another term called the electron transport chain too. I'm not a huge fan of that term. So anyways, we're going to see here that there are these things here called electron transport proteins. I'm just going to write this on the bottom. Electron transport proteins. So what's going to happen is that NADH and FADH2 are going to drop off their payload of hydrogens and electrons. So they're going to release the electrons and also the hydrogens. Okay, now what's going to happen is these electrons are going to be passed from electron transport protein to electron transport protein. We call this the electron transport chain. Now the reason why this is because opposite charged things attract. So basically what happens is that as the electrons are moved from one protein to the other, the hydrogens follow obediently. And as the hydrogens follow, they're actually going to pass through an enzyme called ATP synthase. And this is very important. Now, enzymes are powered by ATP. And this is a special enzyme that makes ATP. So if we had an enzyme that makes ATP, be powered by ATP, it would be like trying to get rich by selling 4 quarters for $1. The alternative is you can find something else to power ATP synthase, and that's going to be hydrogens. So the hydrogens are going to pass through ATP synthase, and as they do, you are going to have ADP converted into ATP. And the ATP can be used throughout the body in order to make or to do things, to move muscles, to transport chemicals, to do reactions. So ATP gives the body energy to do things. Now the very final step is that oxygen, because recall that we do breathe in oxygen, is going to accept the hydrogens and the electrons, like this. When it does so, it's going to become water. And the process ends. So let's recap this again. So NADH and FADH2, whenever they get into the inner membrane space, they're going to drop off electrons and hydrogens. The electrons are actually captured by these proteins called electron transport proteins, and they're moved from one protein to the next, and as they do so, they are going to move along the hydrogens through what's called an electrochemical gradient. Negatively charged electrons drag along positively charged hydrogens. ATP synthase is an enzyme that takes ADP and turns it to ATP. It's powered by the movement of the hydrogens. And this is important because if you did not have the movement of the hydrogens, then you would not be able to power ATP synthase. You'd have to use ATP, which would be pointless. We'd be using ATP to make ATP, as I said earlier. Now, you don't want hydrogens or electrons just floating throughout the body unattended, so they're actually combined with oxygen to form water. So therefore, the entire equation for cellular respiration is glucose, which goes into cellular respiration, plus oxygen, which we breathe in and is in the last step of cellular respiration. It's going to give us ATP, indeed, CO2, which was lost during the citric acid cycle, and water, which is created at the very end of cellular respiration. And these are energy pathways. I know, congratulations. So we'll take a quick break, then we'll be back with the next part. Okay, everyone, now we're going to talk about no, that's actually later, sorry, parts of a neuron. And specifically parts of a neuron that we call a motor neuron. So we'll be talking about this a bit more, but there's really two types of neurons here, well actually three. Sensory neurons, and as I said, not going to go into this too much. Sensory neurons, what they do is they have the enviable job of delivering information to the brain. Then we have interneurons, and these are neurons that comprise the brain. So they are found exclusively in the brain. Then we have the last one, motor neurons. And these deliver electrical impulses, which you'll learn about next, to muscles to contract. So basically how it works is that this is the brain, and as I said, we'll be talking about this later on, is you have sensory neurons. They deliver information to the brain processes it, then gives an action through a motor neuron to move some kind of muscle, either voluntarily or involuntarily. All right, so now we're going to do about the parts of a motor neuron. Sorry for that brief pause there. So now we're going to talk about the parts of a motor neuron. All right, so the first thing that we're going to talk about is, so let me draw before I get too excited, tell us what we're going to talk about. Let me draw neurons first. So this is a typical motor neuron. And you'll see that I'm drawing this large circular thing. It's actually something that we call the cell body. Go ahead and label this. So this here is called the cell body. Cell body. And that's actually the living part of the neuron. As long as nothing happens to the cell body, the neuron will live. However, we'll talk about in a moment what happens to something happens to the other part of the neuron. So anyways, this part here of the neuron is going to be called the dendrites. What the dendrites do is they actually receive information from adjacent neurons. So dendrites receive a signal. So they receive information. And the cell body, when it receives the signal, it's going to pass through the cell body and it's going to run down a long tube called the terminal axon. Actually, this part's just the axon, sorry. This is called the axon. So it runs down the axon and then it's going to get to the end of the axon called the terminal axon. Now you may recall in the last chapter how we talked about how calcium channels open, calcium goes into the dendrite, so on and so forth. That part happens here. So now we're actually getting to the structure where we're talking about the process of how that signal actually gets to the end of the terminal axon and allows for a neurotransmitter to be released. So anyways, what happens here is that the motor neuron is going to receive information from the central nervous system for what to do. Now, what's going to happen in the part we talk about next is that we are going to generate an action potential in the cell body. It's going to run through the axon to the terminal axon. Now, one thing that is important about motor neurons is something called myelination. Myelination. So normally, here is how the information would get from the cell body to the terminal axon. So just to summarize, the dendrites are going to receive information from an adjacent neuron, and that information is going to be received by the cell body, sent to the cell body. Then it needs to go from the axon to the terminal axon. Now, usually that signal is going to go down like this, going to travel in like a wave, which is pretty fast. But there is a structure on top of neurons that is sometimes there called myelin. We'll talk about that later. It's actually a fatty substance that is secreted and wraps around the axon. And what happens here is it actually allows the impulse to bounce, which gets to the terminal axon a lot faster than if it were just to travel. So more or less, the more myelinated an axon is, the faster the impulse will move. This is important for very long neurons that need to get information for parts of the body that are far away from the brain. Which is pretty interesting to think that your brain generates a signal, for example, your pinky toe to wiggle, and notice how fast that happens. That literally happens the speed of as fast as electricity can go. So it's pretty neat. It's a pretty neat system that we have in order to create muscle movement. All right, so next we are going to watch about action potentials and how neurons actually generate electricity. And then we're going to go into lever systems. All right, everyone, for the next part of the lecture, we are going to talk about generating in action potential. Now, as we talked about, excuse me, I'm just going to finish this by putting gates onto these channels. These are going to be channel gates. So as we talked about is how neurons communicate information is they are going to receive a signal of some sort at the dendrites. And then the signal is going to be propagated down the length of the axon using electricity. We call this electrical impulse an action potential, and it resonates down the axon. Then at the axon terminal, you're going to have usually neurotransmitter release. So what you are doing is communicating the signal from the dendrites to the site where the neurotransmitter needs to be released. To show how that happens, we are going to use a cell model. So normally, the cell is negatively charged, or the cell membrane is negatively charged compared to the outside environment. The reason why the cell is negatively charged is because there's lots of stuff inside the cell that has a negative charge. And many cells have a resting baseline. It's called a resting membrane potential of about negative 70 millivolts. So we're going to talk about the stages down here. So the first stage is going to be called the resting membrane potential. I'll just do MP. It's written out more in the outline. And here are the situations. Well, when the cell is at resting potential, the sodium channels are closed. The potassium channels are closed. And the sodium-potassium pumps are doing their thing. What they are doing is they are going to move potassium into the cell, and they're going to move sodium out of the cell in a ratio of 3 to 2, 3 sodiums for 2 potassiums. Now, because of the actions of the sodium-potassium pump, you're going to have most of the sodium outside the cell. Going to draw some sodium. I'm not going to go nuts drawing the sodium. And most of the potassium is going to be inside the cell. And you might be thinking, well, what's the point of this? They both have the same charge. Well, the point is going to be concentration gradients, as we'll see. So both channels are closed, meaning that once sodium and potassium is in its respective environments, it's not going anywhere. You may recall the book talking about potassium leak channels. I'm going to conveniently ignore those just because it's going to complicate an already complicated topic. So let's just get down the get down the foundational knowledge. And the Na plus K plus pumps, one thing to keep in mind, they're always operating. So even though I'm putting down operating, that's only formality because they're always operating. Always moving out 3 sodiums for 2 potassiums. Now, whenever a neuron needs to fire or propagate a signal, it needs to generate an electrical impulse along the membrane. Now, you have to ask yourself, how can we make, in this situation, how can we make the cell more positively charged on the inside compared to the outside? Well, let's brainstorm some ideas. Let's say that I decide that I'm going to open up the potassium channels. I'm going to take my eraser here. We're going to open up the potassium channels. And if I do that, you might be thinking, well, that would work. Well, let's think about that. If we take off the gates for the potassium channels, potassium is going to go down its concentration gradient, because recall that channels have to be passive. It's going to go down its concentration gradient, and the cell would become more negatively charged compared to the outside. Instead of our curve going like this, which we want, it would go like this, which we don't want. All right, so that's a bad idea. But what if we take off the channels, or we take off the gates off the sodium channels? Well, guess what? We have a very steep concentration gradient of sodium outside and potassium inside. So what's going to happen is the cell is going to go through the second stage, which is called depolarization. It's going to reverse polarity, depolarization. What that means is that the cell is going to have a positively charged electrical environment. The inside of the cell is going to have that positively charged environment compared to the outside. What's going to happen is that sodium channels are going to open, and sodium is going to flow into the cell, like this. So now we haven't lost any potassium ions, but we've gained sodium ions. Because of that, we are going to have the cell membrane is now going to become positively charged compared to the outside environment. It's going to go up to 0, and then it's going to go up to 30. We call that depolarization. The cell is now going to generate an electrical impulse. As soon as it gets to plus 30 millivolts, we're going to have that electrical impulse being propagated down the axon like this. Once it's generated, it's off to the races. But we don't want it to go above plus 30 millivolts. But we'll get to that in a second. Let's just finish our flow chart. So this time, Na plus channel is open. Then we have potassium channels closed still. And the sodium potassium pumps are still operating, but I'm not going to put that down. The cell is going to become positively charged. I do want to note, though, it is very important that the impulse passes what's called a threshold point. So this here is going to be the threshold point, around 0. I'm going to throw it in dots. If the electrical impulse does not exceed this threshold value, it's not going to depolarize. We call this a gosh, why am I thinking on the term? This is, oh, sorry, I was going to say gated. It's graded. It's called a graded potential. I'm going to write that down. A graded potential is a change in voltage without depolarization. So think of it like this. If you touch the desk, you are going to create a graded potential in your nociceptors, your pain receptors. But it's not going to be sufficient stimuli for it to depolarize. Now, if you decide to, in frustration, to punch your desk, don't do that, then you are going to definitely generate enough electrical activity to depolarize those nociceptors. So that's an example of graded potentials. It's like kind of turning it on, but not quite. Like my dryer, for example, if I just touch the button a tiny bit, it'll start to spin, but it won't actually turn on. If I hold the button, though, it will turn on. A graded potential is like when you just tap the button and it just spins for a second and then it stops. So a graded potential, you do not get depolarization. If it exceeds what's called the threshold point, then you will have depolarization, and the threshold point is in yellow. All right, well, the issue is that now we don't want the cell to be positively charged anymore. It's done its job. So now we need to do what's called repolarization. We want the cell to rapidly become negatively charged again. We call this repolarization. Repolarization. So what we're going to do now is we don't want sodium to come in anymore. So what we're going to do is we're going to close the sodium channel. keep in mind the sodium is still in the cell, but now it's just the channel is going to close, so no more sodium can come in. So the sodium channels are closed. And what's going to happen is now the K plus channels, the potassium channels, are going to open. And the sodium potassium pumps are still going strong. So now we're going to take away the channels off the, the gates off the potassium channels. What's going to happen is potassium is going to go down its concentration gradient. It's going to exit the cell. So now we're going to have a bunch of potassium go outside the cell. Okay, so we're going to lose some potassium. Now what's going to happen is that this is going to result in something called hyperpolarization. So it's going to go down to, instead of negative 70 millivolts, it's going to go down to about negative 85 millivolts. And that's going to be called, I'll put that here. Let's see, where should I put it here? Hyperpolarization. Cannot write today. Polarization. What this is that the membrane potential becomes more negative than baseline. And the reason why is because, as you can see here, the environment of the cell is in disarray. The ions are in their wrong places. The cell ideally wants potassium inside the cell and sodium outside the cell, so we can have a strong gradient that can result in a strong depolarization. Right now, the ions are kind of mixed and matched. So the reason why hyperpolarization occurs is because right now the cell is not ready to depolarize again. It needs time. It needs time to restore the ions to their correct locations. So the purpose of hyperpolarization, recall that a cell will not depolarize until it passes this threshold value. The purpose is to make it quite a bit harder for that threshold value to be passed by. So it's a protective mechanism that allows the cell to reach resting membrane potential. And guess what? That's exactly what happens next, is we have a return to baseline. So now what's going to happen is we are going to go right back to return to baseline, right back to resting membrane potential. So we see here that at resting membrane potential, sodium channels are closed, potassium channels are closed, Na plus, K plus pumps are operating. Let me return the cell to resting membrane potential. Give me one moment here. No, go back. Thank you. All right, so I'll feel better once I get the cell back to where it should be. Okay, great. Let's put some gates on it. So this is how the cell looks at resting membrane potential. When depolarization happens, as we know, the sodium channels are going to open. Sodium is going to begin to go into the cell. It's going to reach positive membrane voltage because now you've added positively charged ions to an environment that already has positively charged ions. Recall that the ions don't really matter. It just is about the charge. Once positive 30 millivolts is reached, the action potential will begin to propagate down the axon like this in a wave pattern. And then we want to get the cell back to its negative membrane potential. So now we're going to close the sodium channels, open the potassium channels, potassium goes out, we return the cell to a negative membrane voltage, and then the sodium potassium pumps will have necessary time to restore the cell to its resting membrane potential status. Now, cells also have channels for chloride. And this basically keeps the neuron off. And that's one thing that I believe benzos do. What they do is that they open up these chloride channels to inhibit the neurons from firing, which creates sedation. I put in here in the outline about gated channels. We've already talked about that. These channels are mainly voltage gated. That's how the sodium channels and the potassium channels know to either open or close, is they're mainly voltage gated, especially the potassium channels. All right, and next up we are going to talk about how neurons actually work and types of neurons. So, see you in a bit. All right, everyone, well, welcome back. We're now going to talk about lever systems, which more or less explains the, I guess you can say, physics behind muscle movement. So we're going to use the upper arm and the lower arm as a model for how our muscle systems work. So I'm just going to do the upper arm here. Then we are going to have this part here called the fulcrum. Then we're going to have the rotational, or what's going to move, is called the lever arm. Okay, so this here is going to be the fixed point. The fixed point. The rotational point here is going to be called the fulcrum, which would mimic the elbow. And then this part here is going to be called the lever. And the lever is the part that's being moved. Now, using this model is we're going to talk about two types of lever systems. Now, keep in mind here, the goal is, and if you extend your arm, the goal is to close that angle. Close the angle. And to do so, you need some sort of force to close that angle. And in this case, it's going to be your muscles. So the idea behind muscles is contraction. If you want to close the angle, then you need to have something shorten that angle. So this muscle, what it can do is it can contract and it can close that angle. Now the question is where should that muscle begin and end? Very valid question. Why can't it just happen over here? Why can't it happen like this? Or what about right in the center? Is there a difference? And the answer is yes. So first, let's talk about a lever system called the speed lever. Okay, so what we really want to know is, where exactly should this muscle go? Well, we have a couple options here, but first, let's talk about a wrench. Okay, so I'm going to try and draw us a wrench. Don't expect the creme de la creme here, you know. So anyways, this is a wrench. I think it looks kind of like this. Sure, why not? And this is the handle of the wrench. This little, yeah, I don't know. I think this is a wrench, whatever. Now, let's say that you want to turn the wrench really fast. Where would your hand be? Like you wanted to turn it around the rotational point, which is the fulcrum. You want to turn it really fast. Now, to do so, what you would do is if I were to tell you, turn this wrench as fast as possible, you probably would put the force, which is your hand, and you're going to put it here. And then you're going to spin it around at this point here, and you can spin it really, really, really fast. Now, we call that a speed lever. So a speed lever has these characteristics. It has the force close to the fulcrum. Now, the advantage to that is that you're going to increase speed, but the expenses is going to be decreased force. So what this means is that with a speed lever, it will allow you to close this angle faster, but you won't be able to generate as much force as with the opposite one called a power lever, which we'll talk about in 1 second. Okay, so the speed lever, as I said, is designed to move things quickly at the expense of power. So it's the opposite. Now let's say that you are trying, so once again, if I were to ask you to turn this wrench as fast as possible, you would hold it here, and you would turn it around and around like this. You would spin it with your finger, and you would move it really fast. But if I were to ask you to exert as much force as possible, what you would do is you would actually grab it away from the fulcrum. You would grab it at this point here, which would be the end of the wrench. And by doing that, you could exert maximal force to move or to rotate the wrench around the fulcrum. So if I were to ask you to loosen a really tightly bound bolt, you would pull it from, you would pull it further away from the fulcrum. That's called a power lever. So a power lever, the force is away from the fulcrum. The result is that you lose speed compared to force. Now, the downside also to this is that since the muscle is going to be more like this, what's going to happen is that the muscle actually has to contract further in order to do the same movement. So the problem is that you are going to use more energy. It would be like driving a diesel truck to get groceries when you can do the same thing with a Honda Civic. Sorry, the speed lever is going to use less energy. Okay, now you might be asking, well, which systems are most common in the human body? And the bottom line is that actually speed levers are the majority of lever systems in the human body. And that's because with humans, we have survived mainly because we have the ability to do things quickly without the use of much energy. We are very, very energy thrifty. So most of our levers in our bodies are speed levers. And if you're bored, take your arm and start doing bicep curls really fast. You realize you can do that fast because that you have a speed lever. So that is the lever systems that help most mammals survive. Now, there are some examples of power levers in our body. For example, the calf. the calf that inserts into the heel. It's called the Achilles tendon. And if you look, the calf muscle is pretty far away from the heel. And if any of you have done calf raises at the gym, you realize that you can exert a lot of power, but at the expense of speed. Because if you keep going on your tiptoes and down, it's not going to be nearly as fast as what you could do, for example, moving a finger or curling your arm. So it's a rather small motion that takes more time because it's a power lever. All right, so once again, is the point of a muscle is basically to provide force in order to close or open an angle. So what's going to happen is that you have the fixed point, which in this case would be the upper arm, and you have the fulcrum, which is the elbow, and the lever, which is the part of the body that is pulled on. So for example, the upper arm is where the bicep begins. It inserts into the lower arm, and you move the lower arm whenever you do a curl, for example. And the fulcrum, as I said, is the rotational point. If the force inserts close to the fulcrum, then it is going to be called a speed lever. If it's closer to the end of the lever, it's going to be called a power lever. Most levers in the human body are speed levers, and it's a large part of our survival as a species.